Welcome to this presentation on test crosses and dihybrid crosses. A test cross is used to determine what the genotype is of an unknown organism that has a dominant trait. Recessive traits are always known. Recessives are always little. But a dominant can be homozygous or heterozygous. Here we have a dominant trait, but we don't know what the second allele is. It could be heterozygous if it ends up being a big P, or homozygous recessive if it were a little p. If all the offspring were to turn out purple, then the parent would have to be homozygous dominant. If they turned out half white and half purple, then they would have to be heterozygous dominant. So here we have an unknown genotype of a parent, and we need to cross it with a homozygous recessive. Here's the recessive. If it's homozygous dominant, the results would be 100% purple. So all of the offspring are purple. If the genotype were heterozygous dominant, then half of the offspring would be white and half of the offspring would be purple. So we are able to determine the genotype of the unknown parent using the genotypes or the phenotypes of the offspring. Here we have again an unknown dominant, and it is crossed with a recessive. If the unknown dominant ends up being homozygous dominant, then all of the offspring will be black. If the unknown dominant ends up being heterozygous, then half of the offspring will be black and half of the offspring will be chocolate. Pause the video and try this sample problem. We will always cross a dominant with a recessive. So in this case, a white Dalmatian is recessive and therefore little d. We know that the other parent is dominant, but we don't know what the second allele is going to be. It tells us that 3 of the 5 puppies came out white and 2 are spotted. So if we produce the Punnett Square for this, in order to get white offspring, what does this allele need to be? If we think about it, it needs to be a little d. Therefore, the unknown parent's genotype would be heterozygous dominant. Now I'm going to talk about dihybrid crosses. These are used to show the possible combinations of 2 characteristics or 2 traits. I'm also going to talk about linked traits, and they tend to stay together because they're very close on chromosomes. So Mendel came up with the law of independent assortment. He said that traits are distributed to gametes independent of each other. In other words, during meiosis, crossing over separates traits from each other, and the resulting gametes will all be different.
We can use a diploid cell with just 2 pairs of chromosomes to illustrate a genetics concept called independent assortment. We will track the movement during meiosis of the cell's 2 pairs of homologous chromosomes. Before meiosis begins, the cell synthesizes new DNA and thereby replicates each chromosome. Early in meiosis, the pairs associate. Note that the chromosomes in a pair are not quite identical. In one pair, for example, one chromosome carries a recessive allele of a gene, such as a little s, and the other carries a dominant allele, such as a big S. The two pairs line up at the cell's midplane during metaphase 1 and may adopt one of two alternative alignments. The alignments provide the basis for independent assortment. The cell may adopt alignment 1, in which the little s and the big Y alleles end up in the same daughter cell. Or the cell may adopt alignment 2, in which the little s and big Y alleles end up in separate daughter cells. From a large pool of cells that initiate meiosis, on average, half of them adopt alignment 1, and the other half adopt alignment 2. During the second half of meiosis, the two cells from each alignment divide into 4 haploid cells. The result is an equal number of the possible genotypes: little s, big Y, big S, little y, little s, little y, and big S, big Y. Because there are equal numbers of genotypes, we can say that the alleles of the S and Y genes assort independently during meiosis. Note that alleles of different genes always sort independently if the genes reside on different chromosomes, but not necessarily if they reside on the same chromosome.
There is an exception for linked traits. So normally, organisms go through crossing over. And when this happens, traits can be separated from each other. And they can be different. But with linked traits, they're close together on the chromosome. As seen here, A and B are very close on the chromosome. So they will tend to stay together. They are called linked traits. Here's a dihybrid cross. In a dihybrid cross, the first thing we need to do is figure out what the possible gametes are for the female and for the male. And I will tell you how to do that. The best way to do that, once we know the parent, is to do FOIL. So the possible gametes are going to be the first allele for each, which in this case would be big R, big Y. Then the outer, which would be big R, little y. The inner, which would be little r, big Y. And the last of the two alleles, little r, little y. And once we know that information, we can set up the dihybrid cross and carry the letters down and over like we do in a normal Punnett Square. So again, pause the video and do this practice problem. So it wants us to figure out the gametes for an individual that is homozygous dominant for seed shape and heterozygous dominant for seed color. Well, seed shape is represented by the letter R. If it's homozygous dominant, it's going to be big R, and heterozygous dominant for seed color would be big Y, little y. So if we look here, this is the genotype for the parent. Using FOIL, they were able to get the 4 possible gamete combinations from the one parent and the 4 possible gamete combinations from the other parent. This practice problem we will do together. In summer squash, white fruit color, capital W, is dominant over yellow fruit color, lowercase w, and disc-shaped fruit, capital D, is dominant over sphere-shaped fruit, lowercase d. If 2 squash that are both heterozygous for both traits are crossed, what will the phenotypic and genotypic ratios be? So the first thing we need to do is identify the parents. And in this case, it tells us that both are heterozygous. So big W, little w, big D, little d, is crossed with the same parent. Now we need to figure out the 4 possible gametes. And to do this, we use FOIL. So we have our first big W and big D. Our outside big W, little d. Our inside little w, big D. And our last little w, little d. We line these up along the top. And we have the exact same parents, the exact same gametes on the other side. And before we move on, something I want you to notice, the letters are always in the same order. The W is always before the D, even though we have a little w before a big D. It doesn't matter, as long as the letters are always in the same order. So to finish off the Punnett Square, we just carry our letters down. Here we have a big W, and here we have a big W. And then we have 2 big D's. I'll let you finish that on your own. And then identify the phenotypic and genotypic ratios and write them in the correct fashion with the colons. And we'll finish discussing this in class.